Good afternoon. Welcome to everyone. My name is Adil Parucha. I'm a gastroenterologist at Mayo Clinic in Rochester and president-elect of the American Neurogastroenterology and Motility Society. I'd like to welcome you all to this evening's ANMS Virtual Symposium, which is part of our ANMS Basic Concepts in Neurogastroenterology. And I'd just like to put in a plug for all of you that are not members to please consider becoming members of the society. There is a lot that we do for you, for our patients and the field. Before we get started, I'd like to remind you all of our upcoming ANMS clinical meeting on July 26 to 28, 2024. This year, it's in Baltimore, Maryland, and we're putting together an exciting program. So switching gears, moving on to today's virtual symposium, which is entitled Modeling Enteric Nervous System Using Organoids to Study Motility and Functional GI Disorders. Our moderators are Dr. Robert Heuckeroth and Brian Gulbransen. Dr. Heuckeroth is a pediatric gastroenterologist who specializes in looking after children with really serious bowel motility disorders. And he also happens to be a developmental biologist, a cell biologist, and a biochemist who studies fundamental mechanisms underlying bowel motility. His group discovered literally dozens of genes that impact ENS development or function, gene-environment interactions in the ENS, and he has ongoing basic and translational research studies focused on visceral myopathy, Hirschsprung's disease, and pseudo-obstruction. And he does this all by leading the Susie and Scott Lustgarten Center for GI Motility at the Children's Hospital of Philadelphia, and he's especially proud of the wonderful trainees who have worked with him in the lab in the hospital, many of whom have actually joined the call today. Dr. Brian Gulbransen is a Michigan State University Foundation professor in the Department of Physiology at Michigan State, and also we're fortunate to have him as a counselor of the ANMS. He did his bachelor's degree in zoology and physiology from the University of Wyoming, his doctoral training in neuroscience at the University of Colorado, postdoctoral training at the University of Calgary, before joining the faculty at Michigan State in 2012. His research focuses on the mechanisms of neuroinflammation in the intestine, and he has a specific passion for understanding the role of enteric glia in health and disease. And indeed, his work has uncovered some of the fundamental rules that govern intercellular communication between glia, neurons, and immune cells in the gut. Dr. Gulbransen is currently supported by grants from NIDDK and a PPG from NHLBI that focuses on interactions between adipocytes and nerve fibers in perivascular adipose tissue. So with that, I'll turn over the program to Drs. Heuckeroth and Gulbransen. Thank you again for joining this seminar.
Thank you very much for that great introduction, Adil. And thank you all for joining us tonight for this great symposium. We have two excellent speakers. And just before we get started, I want to remind everybody that questions should go in the Q&A box down at the bottom of your screen. And those will all be answered during the Q&A period at the end of the session. So to start us off, it's really my pleasure to introduce Dr. Faranak Fattahi, who is an assistant professor in cellular and molecular pharmacology at the University of California, San Francisco. So the Fattahi lab pioneers the use of human pluripotent stem cells to create intricate models of the peripheral nervous system. These models integrate diverse cell types and stimulation techniques, enabling in-depth studies of complex peripheral neuropathies and applications in drug discovery and regenerative medicine. So we're all really looking forward to your talk tonight, Faranak, and the presentation is entitled Derivation of ENS Models from Human Pluripotent Stem Cells for Modeling and Treatment of Disorders of Gut-Brain Interaction. So Faranak, go ahead and take it away.
Thank you very much, Brian. I'm delighted to join you all tonight, tonight my time, because I'm currently in Dublin, to tell you a little bit about our work on using pluripotent stem cells for modeling disorders of gut-brain interaction. So my lab primarily uses pluripotent stem cells to generate models of peripheral nerves, with a focus on the nerves inside the gut, which I'm going to get into in a little bit. But the rationale for that is these nerves are highly inaccessible from primary tissue. So, you can use mouse models to, study how they work and how they interact with their target tissues. But because a lot of these are not accessible in large numbers, we have issues with being able to isolate them in sufficient numbers to do high throughput assays, drug screens, or genetic manipulations in vitro. So as an alternative approach, we develop organoid models to create various different components of the peripheral nervous system. And our goal is to use these models to understand how they develop, how they interact with their target tissues and organs, and how they're affected in different kinds of disorders. And once we have a better understanding of the disease mechanisms, we can use these systems to do drug screens and come up with therapeutic strategies. So I'm going to focus on one of the primary areas of research in our lab, which is the disorders of gut-brain interaction. And these are formerly known as functional GI disorders or enteric neuropathies. They affect the nerves in the gut, but they could also be, occurrences or manifestations as a result of various different neurological diseases like Parkinson's disease, autism, Alzheimer's disease, or they could be induced by metabolic problems, side effects of drugs. So collectively, all of these affect the enteric nervous system, and they're very, very common. But unfortunately, we don't have good ways of managing them and treating them in patients. And one of the reasons is because the affected tissue, which is the enteric nervous system, is really challenging to study and has been challenging to study for a number of decades now. So, disorders of gut-brain interaction is an umbrella term, but depending on where in the GI tract it manifests, it could take different names and has different manifestations. But a lot of times these are linked to dysfunction of different components of the nervous system, namely enteric neurons, where these neurons do not function properly. Maybe they don't produce sufficient amounts of neurotransmitters. Maybe there's a disbalance in the neurotransmitter production, and maybe there's degeneration of these neurons that cause these dysfunctions in different components of the GI tract. So in order to study them, what we do, as I mentioned earlier, is we model the process of fetal and embryonic development in the lab and create these models, organoid models from stem cells. And here's just a picture of what these models look like typically. Here we're looking at neurons in red and glia are shown in green. So these models that we create from stem cells are a combination of various different kinds of neurons and glia. And I'm not going to have time to get into the details of this, but a lot of what we do is focused on figuring out how well they represent the actual primary tissue. Do they match with their primary counterparts, both in terms of molecular features and also their functional features and their ability to recapitulate disease phenotypes? So one of the key features that we want to make sure that our models have is their ability to recapitulate the diversity of cell types inside the ENS. So here we're looking at a number of different markers of various different neurotransmitter subtypes of neurons. So here is just immunofluorescence imaging, looking at multiple markers for different neurochemical classes. And as you can see, our stem cell derived models shown here on the left are a combination of different kinds of neurons that reflect the similar diversity to what you see in mouse and human primary tissue. So what this means is we have now access to these models, these neurons, and I encourage you to take a look at our preprint that gets into the details of characterization of these different kinds of neurons. But I'm going to talk a little bit more about how we leverage these models to study disease. So the first thing I wanted to talk to you about is a work that was recently published by a postdoc in our lab, Dr. Alireza Majd. What he wanted to do was take a genetics approach to look to see which one of the neuronal subtypes in the ENS are selectively vulnerable to DGBIs. So he looked at the UK Biobank, looked at 5 different categories of DGBIs, and he ran GWAS studies on all of these 5 categories. and then comparing the list of genes that, or variants that show up as significantly correlated with incidence of DGBI with single-cell RNA sequencing data sets that are publicly available, both from primary tissue and our stem cell-derived models, it was able to show first that the majority of these disease susceptibility genes are expressed at high levels in enteric neurons compared to other components of the GI tract. And this was not surprising, but was reassuring to see that a lot of these disease susceptibility genes are neuronal, and that this is in alignment with other findings in the literature on the basis of these pathophysiologies. But going one level deeper into cell type specificity of the expression of these disease genes, we looked at expression level of these susceptibility genes in various different neuronal components in our models, again, and publicly available primary ENS data sets. And here's just one example from one of the five different DGBIs that we looked at. And as you can see, some neuronal subtypes have higher degree of expression of these disease susceptibility genes. And among these highly susceptible subtypes of neurons, we see inhibitory neurons. Here, for instance, nitrogen neurons show up as some of the highly more susceptible cell types to these different disease conditions. And that's, again, in alignment with what others have previously shown to be the case, that inhibitory neurons seem to be more vulnerable to disease. So this data is now publicly available on our website under this database called BBN browser. You can input your genes of interest and look to see the association of the different variants in those genes with different categories of DGBIs. And we're working on improving this database, adding additional GBA studies from other data banks and also other components of the peripheral nervous system. But as we know, and as I mentioned in the beginning, we know that a lot of DGBIs are not caused primarily by genetic drivers. There may be some susceptibility genes, but a lot of the causal known factors in DGBIs are things like, as I mentioned, metabolic conditions, or in this case, what I'm going to talk about, exposure to specific medications. So we know from various studies in the literature that a specific class of chemotherapy drugs called platins have very specific side effects on the enteric nervous system. So a former PhD student in the lab, Dr. Michaela Richter, took on this project where she used our stem cell-derived models to model the effect of these chemotherapy drugs on the enteric nervous system. One of the first things she tried to do was look at comparing our enteric nervous system models to other models of the peripheral nervous system. Here, she's exposing these different kinds of neurons to different classes of platinum chemotherapy drugs. And as you can see pretty clearly, enteric neurons have a higher degree of susceptibility to these chemotherapy agents. And this cell death and cytotoxicity is accompanied by other hallmarks of stress, including DNA damage, which is the primary mechanism of these chemotherapy drugs, oxidative stress, and neurite degeneration that ultimately leads to excessive stress and cell death. So using these phenotypic hallmarks, she performed a high-throughput pharmacological screen with the goal of identifying mechanisms and pathways that are involved in this cytotoxicity. From analyzing the drug targets and putting together this pathway analysis, network analysis, she found that the specific pathway linked to muscarinic cholinergic signaling through acetylcholine receptors is actually responsible for specific cytotoxicity in our enteric neurons. She found that when she blocks this pathway, this GQ-GPCR signaling pathway at different levels of the signaling pathway, she can actually rescue this disease mechanism. So here, as an example, blocking this calcium channel is capable of a full rescue or full prevention of cytotoxicity in enteric neurons that are exposed to platinum. So if there's excess activity of this signaling pathway, this buildup of calcium should result in hyperexcitability or excessive excitotoxicity in the cells. And that's exactly what she validated next, which is shown here. We use multi-electrode array analysis to look at firing rate in our neurons. We see that when neurons are exposed to platinum chemotherapy drugs, there's a higher degree of electrical activity in the neurons, and that effect goes away when the cells are simultaneously treated with the platinum drug and the HIT compound, which is an inhibitor of this muscarinic receptor. So to further validate the role of this pathway, she performed this genetic experiment where she first looked to see which one of these receptors are expressed in high levels in her neurons, and CHRM3 and CHRM5. She showed that those are the primarily highly expressed muscarinic cholinergic receptors in these neurons. She knocked them out individually and in combination, and she found that individual knockout or a combination knockout of these receptors is actually sufficient to rescue this chemotherapy-induced cytotoxicity in enteric neurons. So far, what I've shown you is just general cultures of enteric neurons, but what about subtype-specific susceptibility? So the next thing she tried was zooming in one level deeper into the different categories of enteric neurons using single-cell RNA sequencing and the prevalence or expression level of different components of this muscarinic cholinergic signaling in the different subtypes of neurons. So here we're looking at, in this data set, 19 different categories of neurons, and looking at expression level of different families of proteins in GQ-GPCR signaling, and subtypes of neurons that have high expression of these pathway components happen to also be these inhibitory neurons that we saw earlier in our genetic data. So here's just some example pathways that were upregulated in these more susceptible neurons, and that we showed that these pathways are reversed when the cells are co-treated with platinum chemotherapy and the anticholinergic drug. And in the less susceptible neurons, that effect is not to the same degree. So this was all predictions based on computational data. She wanted to validate this experimentally, and here what we're looking at is exposing our enteric neuron cultures to chemotherapy drugs and looking at the different degrees of toxicity in different subtypes of neurons. On the left, we have nitrogen inhibitory neurons. On the right, we have excitatory neurons. And as you can see, these inhibitory neurons, nitrogen and GABAergic, have a higher degree of susceptibility to platinum chemotherapy exposure. Is this accompanied with functional changes? So the next experiment she did was looking at this neurotransmitter release assay, where we can measure the amount of neurotransmitter that's released by the neurons in the media. So in this case, we're measuring nitric oxide in our inhibitory neurons. And she found that when neurons are treated with chemotherapy, there's a higher level of production of nitric oxide, even in the absence of external stimulation. And then when the neurons are stimulated, that effect is actually higher. So we actually see that exposure to chemotherapy induces excessive neuronal activity and release of neurotransmitters. Is this relevant to in vivo? So she did 2 experiments here. The first one was to validate this observation in human patient samples. So we got samples from gastric cancer patients that underwent chemotherapy. We saw that when patients receive chemotherapy, there is skew in the proportion of inhibitory neurons versus excitatory neurons. You can see here the proportion of inhibitory neurons is actually reduced in patient tissue samples that receive chemotherapy, and the proportion of excitatory neurons is actually increased. Is this linked to any changes in the function of the GI tract? So to answer that question, she did an experiment in mice. Here, she co-treated wild-type animals with chemotherapy and her hit compound from the screen, which was a muscarinic cholinergic receptor inhibitor. She found that treatment with chemotherapy leads to this constipation phenotype where the total GI transit time is actually increased. When she treats the mice with the muscarinic cholinergic receptor inhibitor, that also leads to this constipation phenotype because she's dampening the overall neuronal activity, but co-treatment actually gets the mice to behave normally. And this is accompanied by similar changes that we observed in human tissue, and this time in mouse tissue, where the proportion of nitrogen neurons is decreased, and the proportion of excitatory neurons is actually increased. And this effect is rescued by co-treatment with the hit compound. So to summarize this part, we see that chemotherapy leads to a selective loss of inhibitory neurons. We believe that this loss leads to an imbalance in the neurotransmitters that leads to a constipation phenotype. Then we expose these mice to anticholinergic drugs, muscarinic receptor inhibitors. The same effect occurs, but this time due to dampening of neuronal activity, but co-treatment, because this dampening of firing activity protects the neurons from excitotoxicity, restores the balance between the neurotransmitters, and leads to a rescue in animals. So I talked about how we can model genetic susceptibility or non-genetic forms of neurotoxicity and enteric neuropathy, but can we use our stem cell models to also come up with therapeutic strategies for treatment of DGBIs? So I mentioned in the beginning that loss of a neurotransmitter production capability or loss of neurons, full degeneration, could be the underlying causes of DGBIs. So the two strategies that we wanted to test is, can we, in diseases that are linked to abnormal or insufficient production of inhibitory neurotransmitters, can we boost the production of these neurotransmitters using pharmacological interventions? And in diseases that are linked to degeneration of the neurons, can we replace these missing neurons using cell therapy? So to just give you a couple vignettes on these two different strategies, this next slide is about like a summary of our pharmacological approach, where we performed a high-throughput screen using a library of neuromodulators, looking for neuromodulators that boost the production of nitric oxide in our enteric neuron models. We found a number of different classes of neurotransmitters that can stimulate production of nitric oxide in these models. And we're in the process of validating these further, but here's an example of how we can validate these in vivo systems. So here, for instance, we're recording, we can measure the changes in the pattern of motility. As you can see, the bottom sample showing increased levels of motility. And because many of these compounds are FDA approved, we're hoping that after further validation, we can accelerate the translation of these into potential clinical applications in patients that suffer from DGBIs. Now, to just give you a summary of the cell therapy approach, similarly, using stem cell-derived models, the idea here was to use a mouse model of this motility that has a deficiency in inhibitory neurons. Here we engrafted human cells that are shown here in red in colonic samples in these mice. And 8 weeks later, we're looking at the prevalence of these human cells, the survival and spreading of these human cells inside the colonic tissue. And as you can see, all throughout the sample that we analyzed, we see a lot of human cells engrafted and survived, and they appear to be homing into the actual ganglionic tissue. And we're now in the process of addressing whether or not these lead to functional rescue in these animals. Here's just an image of neuronal fibers, human fibers shown in red, extending into the muscle tissue. And this is some preliminary data indicating that mice that receive these transplantations have an improved GI transit time compared to mice that have this deficiency in inhibitory neurons, and that this effect is actually, it appears to be higher if the mice receive ganglioids or stem cell-derived cultures that are enriched for these inhibitory neurons. So to summarize, our work on using pluripotent stem cells has multiple components. We develop models that recapitulate the process of fetal development. We use these models to understand how specific subtypes of neurons are specified during development, what regulatory programs lead to the emergence of different types of neurons. We use them to model disease and perform high-throughput screens to come up with therapeutic strategies, and we use them for cell replacement therapy and regenerative applications. With that, I'll just say thank you to everybody in my lab who's been contributing to the work. I mentioned Alireza Majd, Homa Majd, and Michaela along the way, Michaela Richter, and our collaborators, specifically our collaborators at Stanford and at UCL contributed to a lot of the mouse experiments. And of course, our funding support. A special shout out to California Institute of Regenerative Medicine for supporting both our drug discovery and cell therapy programs. With that, I'm going to hand it back over to Brian.
Wow, thanks for a fantastic talk, Faranak. That was really great. So I'll just remind everybody, put your questions in the Q&A. We're going to have the Q&A period after we're done with the second talk. And we'll just go straight in, and I'll turn it over to Bob Heuckeroth, who's going to introduce Maxime Mahe, our second speaker.
All right, it's my pleasure to introduce Maxime Mahe. Maxime is a associate professor at INSERM and an adjunct assistant professor at Cincinnati Children's Medical Center. He obtained his PhD working with Dr. Michel Neunlist at the University of Nantes, and then worked with Dr. Malcolm Heuckeroth and Dr. Jim Wells in Cincinnati. In his own lab, he's been building human bowel models from stem cells, and also incorporating neural crest cells into the bowels to model how mechanical factors influence development and function, how microbes and nutrients, and how incorporating enteric nervous system cells influence the activity of other cells in the bowel. And so, Maxime, I'm really excited to hear what you have to say tonight. Thank you for doing this.
Yeah, thank you, Bob. Thank you for the nice introduction. So let me share my screen. Oops. So yeah, so as Bob said, so I'm an assistant professor at INSERM, and today I'd like to at least demonstrate that we can use now iPS cells to model intestinal function and at least the development of the ENS and its function. So our lab is really interested in understanding how the gut matures and develop over time, so during the fetal development, but also after parturition. And to address these different question, over time, we turn to use a human model of the gut. And for a little bit over 10 years now, we have been diving into the use of iPS cells and ES cells to derive specific gut models. So before going into details in how we can generate gut organoids from iPS cell, I'd like to give you a really, really short introduction to GI tract development, as this is the first basis of our how we actually create and model these organoids. So as you know, that after gastrulation, we have 3 germ layers that arise in the embryo. The endoderm that will give rise to the intestinal epithelium, the mesoderm that will mostly give rise to mesenchyme, and also vascular and muscle cells, among all, and an ectodermal contribution, which are from the vagal nerve crest cells that will give rise to the enteric nervous system. So as we know pretty well how those different cells developed and the gut is actually building during the development, we need to know how we can translate those developmental sequences onto human pluripotent stem cells. And we have been fortunate to find different ways to generate or to reproduce part of the developmental sequence to form the gut. And to do so, we use a human pluripotent stem cell. We generate a definitive endoderm, and we'll pattern the gut using different signaling pathways, using FGF4 Wnt, so that we can generate what we call spheroids. And those are a representation of the primitive gut tube that you find during development. And then we culture those little spheroids in matrigel, and we'll let them grow for a couple of weeks so that we can get what we call the HIO, or the human intestinal organoid. And we can follow those different stepwise differentiation process. We can look at what's happening, all the molecular and cellular sequences that are happening during development. But at the end, we generate an organoid that grows from hundreds of micrometer to a millimeter in size. And it takes about a month to generate this structure. And if we look closer at how this organoid is made of, you can see in the central lumen with an epithelium outside, and there is a supporting mesenchyme outside. So you will appreciate, even though we are generating a definitive endoderm, we do see mesenchyme forming. And if we look at different lineages, and here on the epithelial lineages, we find the enterocytes, the goblet cells, Paneth cells, and endocrine cells as well. So starting from those pluripotent stem cells, we can generate a pretty much a good representation of a human native tissue representing most of the cell lineages. Yet, as we generate those gut organoids from the endoderm, we are not getting what the enteric nervous system. And as you know, the enteric nervous system is originating from the ectoderm, or at least the neural crest cells, delaminating from the neural tubes. And those are pretty crucial cells as they will invaginate the primary gut tube to form the future plexus. So that will give rise to the enteric nervous system. And this is a really critical system to control motilities and different processes in the gut. And this is also a really important system when we regarding to different diseases, including Hirschsprung disease, for example. So our goal was to model the innervation of the gut using the organoid. And what we have been able to devise is to reuse our hPSC-derived intestinal organoid model. We grow them as I demonstrated it before, but in parallel, we were able to generate what we call neurospheres and a neuroepithelium, and using different stepwise differentiation process as well, we can generate vagal neural crest cells. Those are migrating vagal neural crest cells that we can co-culture with those spheroids. We let them grow and we generate what we call the hIO-ENS, so a human intestinal organoid bearing the enteric nervous system. So we can actually dissect it out and study it. We can use different type of iPS cells. This is a representation of an hIO that is growing with a vagal neural crest cell that are bearing a GFP label. And you can see those little crest cells are actually organizing themselves around that hIO structure. If we look closer, this is a control organoid that has not received any vagal neural crest cells. This is the hIO-ENS, and when we look for the presence of neurons, you can see like some of them arranging in the mesenchymal part of the hIO, as well as some glial cells that you can see clearly here. So Elise Loffet and Lisa in Brossard in the lab took on a task to actually characterize those intestinal organoids, and they use a single cell sequencing approach. And you can appreciate when we dissociated those hIO with ENS, a very heterogeneous cell population. So we do have epithelial cells, neural cells, but we do see smooth muscle cells, some vascular smooth muscle cells, endothelial cell, and so forth. So very diverse population of cells with a high heterogeneity. And when we compare that to other data sets of single cell, and especially the Gut Atlas, where we were comparing our hIO-ENS data sets with different developmental tissue from the gut, you can appreciate that the hIO and hIO-ENS reproduces really all marks of the intestinal development, but really early development. So this is really interesting because we can tackle the question regarding development of the gut and the ENS, and this is also an opportunity to understand how that tissue develops and how it could mature over time. So Lisa and Elise went on and thought to interrogate how the ENS would actually influence the intestinal development. And this is a really simple experiment where they compare hIO with hIO-ENS. They use different approaches, including transcriptomics, and they were able to demonstrate that over time you have a separation, a different transcriptomic signature of organoid that have or have not an ENS, and suggesting that over time the growth of the ENS within those organoids are actually changing the signature of this intestinal tissue. And this is just an example of what could happen in the tissue. We were fortunate to see that an upregulation of muscle cell differentiation genes would appear when we would add enteric nervous system in the hIO. And this is an example with the transgenic genes, which is an early marker of visceral vascular smooth muscle development. And we were seeing that every time we would culture hIO with an enteric nervous system, we would see an increase in those markers, suggesting that ENS progenitor cells could pattern the overall mesodermal development. So we went back to the single cell sequencing. This is a comparison between hIO and hIO with the ENS, and you can see that there is like a difference. Other cell types appears over time when you culture the ENS, and they are not necessarily neurons or glial cells. So we looked at the different cell types, and this is again just an example focusing on the smooth muscle cells. We look at the expression of actin G2, and we were able to show that most of the hIO-ENS do have smooth muscle cells that are actually differentiating and suggesting that the ENS is a factor that promotes the differentiation of these cells. So, and this can be also evaluated with different approaches, including imaging. And you do see here, this is just a light check imaging of the hIO-ENS. You do see like alpha SMA positive cells with some early smooth muscle cells that are developing. And this is the GFP or expressing cells, which in this case are the vagal neurocrystal that we have put in the hIO. And you do see that just control hIO don't have any, or at least very little alpha SMA cells or transgenic cells. So this suggests that when you had the ENS in those complex organoid model system, you do see like a emerging cell arising within that tissue. And that illustrates that the ENS is not only essential for the physiology of the gut, but also to have a proper role during development. So we can actually use that model system along with transplantation. So we can use immunocompromised mice. Those are NSG mice. We generate our hIO as I showed you before. We can transplant them in different parts of the animal, but mostly we can use kidney capsule. We can actually insert under the kidney capsule the organoids, and we can let it grow for few weeks, usually anywhere between 6 to 12 weeks, depending on the question you address. The resulting tissue are there. This is an example where you can see like a nice epithelium forming and some laminated structure, including some smooth muscle cells and diverse fibroblasts as well. When we look closer at that type of tissue, the transplantation is indeed really important because it can promote increase the maturation of the organoid so that you can get like a fully developed tissue. Here you would appreciate the inside of a transplanted organoid with the vascularization that is really on the top of some villi. We can do different histology, but here when we compare the tissue with human small intestine, you do see that the resemblance that is really high. And we can look at different cell types. This is just an example with proliferative cells in the epithelium of the gut. You do see the proliferative compartment at the base of the crypt, and this is equivalent to the human small intestine. And finally, bear in mind that that tissue is generated from human iPS cells, and so that we get a 95% human tissue within that mouse model. So when we looked at how it goes, so this is a transplanted hIO on the top of the kidney capsule. You do see how they form. They form like big spheroids, I would say. We can stain for them. You do see like a nice development of the epithelium, partially less developed in the submucosal compartment. When we transplant those organoids that bear an ENS, you do see a development that is equivalent at the microscopic level. But when we section the tissue, you do see like nice epithelial cells, as well as a better developed muscle layer, circular and longitudinal, and we do see some neurons integrated in between those layers. This is also illustrated here by the staining of some neuronal cells that are in between those layer of tissue. When we try to look at the functionality of that tissue, we actually expand the tissue and put that in a dish, and we can realize some electrical field stimulation on the side. You do see like a contraction of the overall tissue, but you would appreciate that this contraction is not only a twitch. We would see like a slow motion happening while we are stimulating the tissue, suggesting that the neurons are actually coupling the muscle to control that contraction. We did many experiments to try to illustrate how that happened, but we were fortunate to show that when we stimulate specifically the neurons in the hIO with the ENS, we would see a response of the tissue, and in this case, a relaxation I won't go into the detail of how we are seeing relaxation, but perhaps we could talk about that later. So you can appreciate that hPSC-derived intestinal organoid system can be used as a model to study the development and the physiology. But you can also appreciate that while they are functional and present in many intestinal lineages, they still have a fetal tissue signature and they have a wrong geometry, right? We are not making tubes, and you know that to study peristalsis, we need a tube. So, and this is the last part of the talk. With Holly Polling and Michael ANMS, we try to devise a way to create tubular intestinal organoids. And we use roughly our method to generate organoid, but we use different scaffolding strategy to get like a longitudinal structure. And this is how it looks like. We can generate those longitudinal structure, and we basically transplant them into the immunocompromised mice. This is the resulting tissue. So here in vitro that we can obtain. So you see like it's more complex, more long, with some epithelial canalization here and some tissue around. And we can actually transplant it into the mice so that we can get a tissue that is slightly more complex. We were really actually surprised to see that. So the tissue is actually engrafting. So this is how it looks like. So with HA, you can get like about a half centimeter tissue, but when you use the groove, what we call the small intestinal grooves, we get like about like 7 centimeters of tissue. And that tissue is actually transplant with higher efficiency than the HA. And you can look at the graft size, and you would appreciate that also the tissue is well developed and probably more developed than the HIU itself after transplantation, with nice epithelial, some laminar layer, including muscles. One really interesting thing that we found is that the tubular organoid also present in ENS. Yet we haven't put any vagal neural crystals, but we do see some neurons that are arising in the tissue. And this is here illustrated with the HNE. You can see like what we usually see in pathology with tissue from patients. You can see like ganglion cells that are located between the muscle. And this is actually tissue derived from iPSC, right? We demonstrated that those are indeed neurons with different staining, but we turn on to test the physiological capacity of that tissue. And this is the tissue you can see here. There are some baseline contraction, but when we do an EFS, you can see that the tissue is actually responding to that EFS, suggesting that there is neurons that are actually engaging in the neuromuscular coupling. Finally, we try to demonstrate that we do have a variety of enteric nervous system cell in that model system. So those are just the HIO. You can see like different cell types. So perhaps it's really small, but trust me, there is different cell types. And when we use the small intestinal grooves, we do see arising some neurocrest cells and some neurons and glial cells that have here because of the label transfer PNS signature. Yet we don't know where those ENS cells come from and how they are arising or actually co-emerging in that tissue. And this is something that we are currently investigating. Well, in conclusion, I hope I was able to demonstrate to you that iPS-derived can be used to study gastrointestinal development and including cellular interaction with the ENS. And those complex organoids are really an opportunity to study human physiology in different ways. And also we believe that it could be a strategy for intestinal tissue replacement, perhaps at the cell level, but why not at the tissue level? I like to finish by the limitation. I think they are important. We have to bear in mind that there are imperfect copy of the reality, and that tissue overall remain fetal or neonatal-like. And again, we still have ways to standardize and actually make better model system with the organoids. I think that the diagram here is the best representation how we can use the iPS and the organoids as far as disease modeling, precision medicine, or why not regenerative medicine. I'll finish with thanking my teams and all the people that have worked in this with those different results that I've shown you, but also my collaborators, Michael ANMS and Holly Polling in Cincinnati Children. And thank you for your attention.
Okay, so thanks for great presentations to you both. So it looks like we've got a number of questions in the chat, and I'm thinking maybe we can go ahead and start with this one from Kent Sanders.
And remember, if you have questions, they need to go into the Q&A box, not the chat box.
Yeah, that's true. Yes, thank you, Bob.
Yeah.
Yep. So the first question, I suppose this could be for both of you. And so Kent comments that he's always worried about how similar the cultured cells are to native cells. And with the human pluripotent stem cells, he's wondering if efforts have been made to compare the expressed gene phenotypes with native cells and comparisons of single cell phenotypes, native versus the pluripotent stem cell models would seem to be a priority. So he's just wondering if you guys could both extrapolate on that a bit.
I guess I could start. So yes, this is such an important focus of, what we do in our lab as we, come up with new differentiation methods. One of the first priorities, as Kent pointed out, is to figure out if they're authentic. And we answer that question with a number of different methodologies, but it seems the emphasis of the question is on gene expression profiles. So in the paper that I referenced by Homamajed et al., that one contains some of the data that we can get into. We didn't have time to get into it now, but we effectively use a number of computational methods now, specifically with access to all the different single cell data sets. We can use correlation analysis, module scoring, unbiased machine learning approaches like single cell net to really look at, in an unbiased manner, if our cells that we generate in vitro have the same gene expression profile compared to the primary tissue. One of the big caveats, though, that we stumbled upon as we were putting this together and as we were revising the paper, because we got the same comments from the reviewers, is a lot of times we don't have a good reference data set when it comes to the ENS, meaning that even the primary tissue data sets that are out there do not match with each other. So when you take primary tissue samples from different sources, different labs, different areas of the GI tract, they have variabilities. There's of course, technical issues with how the cells were isolated, how they were sequenced. So we're not at a point in the field where we have consensus around what the reference gene expression profile for each of the cell types should look like in order to then be able to compare our stem cell derived cells to that reference. So we have some work to do to get to that reference first before we can attempt to do a comprehensive analysis of the stem cell models.
I don't know, Maxime, did you want to say anything about this? Seemed like a very comprehensive answer.
Yeah, I think Faranak said it all, yeah.
All right, we have another question for Faranak from Dr. Lincoln Stamp. Lincoln says, beautiful work. With the rescue of the chemotherapy-induced loss of neurons, affecting the excitatory and inhibitory balance, what is the mechanism? Is this really that you are saving the neurons from dying, or is it possible that you're encouraging regeneration of some of the neurons? I'm paraphrasing slightly.
I see. So inside the tissue, that's something that we haven't really followed up on in great details. In vitro, we are saving them from dying. So we don't see a lot of neurogenesis occurring after a certain point in our differentiations. And we believe that there's lower levels of cell death to begin with, not that there's death and that is followed by another wave of neurogenesis. And then the mechanism of rescue in terms of the molecular mechanism, we think is basically, as I pointed out, there's excessive stress that leads to excitotoxicity. So if we can put a break on the excitotoxicity mechanism through an indirect way, we basically prevent the death from happening, prevent the DNA damage from leading to degeneration and death of the neurons. Hope that answers the question.
Yeah, so
go ahead, Brandon.
No, sure. So did you have something to add, Bob? Sorry.
No,
okay.
I know that's great.
Okay, so so the next question would be for Maxime. So Maxime, Henry Parkman is asking, were the organoids able to be transplanted into other animals or humans to replenish the enteric nervous system or cure disease?
Yeah, that's a good question. At the moment, we transplant in murine models, so including mouse and rats. So we transplant the tissue as a whole, but we haven't found any evidence that the ENS contained in the organoid that are being transplanted migrate through the tissue. And in the contrary, the neurons from the mouse or the rats do not colonize the organoid tissue. The model as it's being made is not a solution for cell transplant. I mean ENS cell transplant, but rather a whole tissue that could be transplanted.
All right, there is a question from an anonymous attendee. Maxime, I think this one is for you. It said, thank you for your talk. Are the emerging enteric nervous system cells in the transplanted tissue from the human stem cells that you started with, or are some of them mouse derived?
Yeah, that's a good and important question. The evidence that we have, it's only human actually. So we looked at different human specific markers, and they are human. So they're coming from the iPS originally. Yeah.
Brian, if you don't mind, I want to take a moment for to ask a question myself. So, I wonder if you could talk, because I think part of what's really beautiful about both of your work is that you start with human cells and generate things that you then sometimes put into mice, and sometimes you're growing them in mice. I wonder if you could talk about the differences between human and mouse biology, and why do you think this is important to be using human cells, and what happens when you put them into the mouse? Is that do you think unusual things happen, that something different might happen if you were putting them into human eventually someday as a treatment approach? That could be for either one of you to think about.
I guess I can start. So, one of the more established differences is the degree of regeneration in the enteric nervous system. There seems to be some evidence that there's some degree of, neurogenesis in the mouse tissue, emergence of new neurons, and it's unclear how much of that is also the case with humans, especially in adult humans and as aging progresses. So that's one difference that could be relevant. In terms of neurochemical properties, we see some differences there too. How the prevalence of co-transmission, for instance, there seems to be some small differences, but again, back to the original issue that I talked about, that we don't have, deep understanding and profiling of both mouse and human in order to really make that conclusion yet.
Yeah, I think we have now the opportunity to tackle those questions, and I believe in, I'm sure Faranak does too, that we have in our hands the ways to directly understand human development and physiology as well.
So Faranak, I have a question here from Rajan Singh from Reno, and what Rajan is asking about is about the diversity of the nitrogic neurons, and so these neurons seem like they're important for a lot of the pathophysiology in the gut, and he's wondering, is there a specific population of these, subpopulation of the nitrogic neurons that's more important, and just how diverse these nitrogic neurons are through the different regions of the gut?
Well, another very excellent question that unfortunately we don't have a definite answer for. So in our initial analysis of our stem cell models and primary data sets, there are definitely subtypes of nitrogic neurons. So just, if you do unbiased clustering of your single cell profile cells in different data sets, you see different types of nitrogic neurons emerge. But how that correlates with functional features, we still don't know. What we do know is there are different degrees of co-transmission. So for instance, one subtype of nitrogic neurons is also GABAergic, another subtype may be also cholinergic. So there's some differences there that we can't yet correspond to specific locations or well-known physiological functions. There's some speculations, but there's no definitive answer, both with stem cell models and I would say also with the primary tissue.
Maxime, I wonder if you could comment for the little organs that you're generating, what stage of human development do they seem to correspond to? Do they look like postnatal tissues or adult tissues, or do they look like fetal tissues?
Well, I think it relates to the first question as well, and Faranak mentioned it. So at least in our hands, from the hPSC-derived organoid we generate when we compare them to atlases, single cell atlases, for example. It seems that we match mostly first trimester, end of the first trimester. So it's very early development. So we are fetal development. The transplantation allows growth and maturation of the tissue, and it seems that we can reach about end of the second trimester, right? So we are trying to devise a way to improve maturation. I think this is the growth, the maturation problems, I would say, are kind of specific to iPS cells. So we are trying to devise ways to actually improve that and reach the adult state. So there is many ways to do that. There is mechanical ways, changing matrices and so forth. So those are important questions, and that we are addressing or trying to address, at least in the lab, and the other group do that too as well.
But do you think you just have to wait a year or two? So, normal gestation is 9 months.
Unfortunately not. I wish. I mean, so we kept them for 7 months and nothing happened, right? So and we transplant for over 2 years as well in mouse, and basically the tissue shuts off. So because there is a lack of vasculature, there is a lack of perfusion with the microbiota, this kind of thing. So we need really to integrate all the major physiological and environmental cues to actually try to reach to that definitive maturation. But there are ways to go.
So there's just something we need to learn about how you proceed to encourage maturation. I wonder, Faranak, one of the things that you said that was, you said a lot of really interesting things. One of the things you said that was interesting and a little surprising to me was that the enteric neuron models seem to be more susceptible to the injury from the platins than the other peripheral neurons that you generated. And I just was astonished to hear that. So do you have some insight into, first of all, how the clinical relevance of that, and also like what the underlying mechanism is?
Well, so clinically, this has, been reported that a lot of the side effects in patients occur in the gut, but there's absolutely other peripheral neuropathies that have been reported. So it's not that the other nerves are not susceptible, it's just probably different degrees of susceptibility. We didn't really look into the mechanisms of, comparing those two categories of neurons in depth to know why the sensory neurons are protected, but we think it probably has something to do, in general, this cell type specific vulnerability has something to do with different coping mechanisms that different cell types have evolved to deal with managing stress and preventing it from leading to cell death. So that's just speculative, but yes.
I love it. I think we are just about out of time, but I want to thank both of our speakers, Maxime, Mahe, Faranak, Fattahi, for giving really spectacular talks, and then for answering all of our questions. If you put additional questions into the Q&A, those will be answered in Doc Matters. And there you go. You have the website for Doc Matter. Again, thank you very much for the speakers and for the audience. Really wonderful to hear about the progress that you guys are making.
Thank you. Thank you all.
Thank you.